And today we're going to start essentially a two-lecture series, 2 lectures on the extracellular matrix and tissue engineering and regenerative medicine. These are not formatted with the pretty UVA slides because this is actually a new lecture that we're working on this class. We had an old ECM lecture and we're kind of revamping it to be a little more engineering friendly and hopefully a little more interesting. I'm probably a really bad judge because I love the extracellular matrix, so of course I'm wildly interested in it, and you guys probably roll your eyes at it. So anyway, all right. So what is tissue engineering and regenerative medicine? So unlike disease states where we talk about prevalence and incidence, here we're just going to address a potential concept in engineering, in particular in biomedical engineering, and that being of tissue engineering and regenerative medicine. So this is ripped right from NIH's webpage, right? So there's the National Institute of Biomedical Imaging and Bioengineering, NIBIB. Yep. Yeah, I apologize for that. I have not gone through that yet. So, so you get less guidance on this particular lecture. We're working on it. All right, so tissue engineering evolved from the field of biomaterials. Biomaterials was this field where, in the classic days, where we were designing metals and ceramics and polymers for things like hip implants, right? And knee implants, these things that were from traditional material science and were being applied in biological context. Anyone want to know does anyone know what one of the first biomaterials on record is? Want to fathom a guess? Starts in one of the most famous examples starts here in this country. No guesses? George Washington. George Washington is thought to have had the first biomaterial on record. He had a wooden tooth fashioned, and they shoved it up in there. Okay. Hardly a biomaterial, didn't last very long. I'm sure a lot of enzymes were breaking that down, but it again is this idea of utilizing traditional materials in biological context. Where tissue engineering has evolved is it's now referring to the practice of combining a scaffold, usually a hydrogel, so a polymer that's comprised predominantly of water, cells, and some kind of biologically active molecules. And we've talked a lot about those. Growth factors, we're going to talk about extracellular matrix proteins, in order to instruct the creation of quote-unquote functional tissues. There are almost no tissue engineering products on the market today. I'll just put it that way. The goal of tissue engineering is to assemble functional constructs that restore, maintain, or improve damaged tissues or whole organs. Skin is probably the best example of something that we've come close to, and there are a number of products in that realm. Okay. So cartilage is another that have been approved by the FDA. I'd like to say that they're as good as they need to be, but they're not. However, currently they have limited use in human patients. They're expensive. It's difficult to get them through the FDA because they're so complex, right? They're not a drug. They're a drug plus a material plus cells plus. And so the FDA has a pretty high level of rigor with which they screen these technologies. So what's regenerative medicine? Regenerative medicine is different than tissue engineering. It's a relatively broad field that includes tissue engineering, but also incorporates this idea of self-healing. So how do we inspire your own body to regenerate itself? What are the inherent mechanisms that you have, and how can we either drug them, right? So introduce drugs, introduce growth factors, introduce biomolecules that help your body remember what it was like when you were a fetus in your mom, right? And growing. So how do we re-engage or reprogram those processes? The idea is the body is using its own systems, again, sometimes with the help of us, with some type of biological material to recreate cells, rebuild tissues and organs. So the stem cell biology field is prevalent in the field of regenerative medicine. Understanding where stem cells come from, how you get them to differentiate down different pathways, right? How how to get a stem cell to turn into a brain cell versus a muscle cell versus a bone cell. I will say that when you're reading the literature, terms like tissue engineering and regenerative medicine have somewhat become interchangeable. They aren't exactly the same, right? So regenerative medicine is a larger field that encompasses tissue engineering as a part of it, okay? All right, so we're going to talk today about the extracellular matrix, and the reason is that what we find, and what I'm going to hope to convince you in a couple of lectures, is that the extracellular matrix winds up being a really nice biologically relevant scaffold material, right? It is your body's biomaterial, right? And it's pretty diverse. It's very, very complicated. We're going to skim, like we're doing in most of this class, across the top surface of this. We'll go to 5 meetings a year with investigators all around the world that all they do is study one protein in the extracellular matrix for the entirety of their career. So it's a pretty complicated thing. It gets 2 lectures in this class, only because I forced it from 1 to 2. Okay. All right. So the broad definition of the extracellular matrix, you do need to know this, is that it's comprised of proteins and polysaccharides locally secreted by cells and assembled into an organized matrix or mesh, into a polymer scaffold, okay? The 2 main classifications are that of what we call basal lamina, or it's also known as the basement membrane, and connective tissue. These are the 2 types. We've kind of talked in the past about 2 types of cells, epithelial and mesenchymal. Epithelial cells tend to interact predominantly with basement membrane. Mesenchymal cells tend to primarily associate with connective tissue. All right. So, and we'll get back to this a little bit more, but this is a pictorial demonstration of that. Here's your epithelium, and they are bound to this basal lamina, or this basement membrane. Underneath that basement membrane, which helps to form this barrier, you have what we call the connective tissue. And these are a fibrillar meshwork, basically a hydrogel system, loaded with cells, capillaries, etc. Here's an SEM image of the basement membrane, right? So you can see it's a very, very thin sheet. These would be the epithelial cells on top, and underneath is the connective tissue. For those that are interested, a lot of these images come out of Bruce Albert's. This happens to be the 5th edition of Molecular Biology of the Cell. He now has a 6th edition. This is like the Bible of cell biology. If you want more resources, it's a really good book to go to. All right, so what are the physiological roles that the extracellular matrix plays? I would expect you to know these. They're pretty straightforward. The primary function is structural, right? It provides structure and structural cues to cells. In the case of both basement membrane and connective tissue, they provide tensile strength. So we talked about the fluidity of cell membranes, right? And so if you can imagine, if I have a sheet of cells with nothing really holding them together other than cell-cell interactions or receptor-receptor ligand receptor bonds, right? I can very easily shear cells apart, right? Because their membranes are simply not robust. What the extracellular matrix does is it provides additional physical strength, tensile strength. In the case of cartilage, it provides the cushioning effect. So your bones are not continually pounding each other, but rather it's a viscoelastic medium that allows you to buffer forces between joints. It can act as a boundary between different tissue types. In the kidney, it actually acts as a filtration system. So there's a very tight mesh of extracellular matrix that actually acts as a way to sieve through molecular species and prevent large proteins, large species from passing into your urine. We'll talk a little bit. So The big ones to know are here on the top, that it acts as a scaffold, that provides tensile strength, cushioning. Again, I talked about it being a molecular filter. We're going to talk at the end about conformational blocking of cryptic sites and sort of this idea, as I mentioned with canstatin, where you have parts of the matrix that live second lives. And we have matrix proteins that look one way, and when forces or enzymes act on them, they can open up, they can shed parts of themselves that go on and serve different purposes. So those are the structural roles of the ECM. Biologically, they direct cell polarity in the case of epithelial sheets. So when I say polarity, I'm talking about what is the bottom of the cell and what's the top of the cell. The bottom of the cell we'll call the basal surface. The top of the cell we'll call the apical surface. And the basement membrane has specific biochemical character which helps the cell determine what's its top and what's its bottom. Okay? The extracellular matrix plays a huge role in cell adhesion. Cells have to grab a hold of the tissue, right? They have to connect to one another, but they also have to connect to the tissue. And so the extracellular matrix is the predominant element in your tissues that cells are grabbing a hold of and holding on to. It plays a huge role in morphogenesis and differentiation, and we'll talk a little bit about that at the end. Migration, proliferation, and prevention of apoptosis, right? So prevention of programmed cell death. Most cells in your body are what we call anchorage dependent, which means that if I remove them from their physical substrate, they will undergo programmed cell death. There's some interesting examples of cells that are not anchorage dependent. Your immune cells would be one, right? Because they have to circulate in your bloodstream. And they actually go back and forth from anchorage dependent when they're in your tissues to anchorage independent when they're in your bloodstream. Anyone think of another cell, very classic cell, that tends to be anchorage independent? Yeah.
Red blood cells?
Well, so red blood cells are anuclear cells. So I will be more specific. Can anyone think of a nuclear cell that's very classically anchorage independent?
Maybe I'm wrong.
It's a guess.
I don't know. Lymphocytic?
So lymphocytic cells are white blood cells or immune cells. So I would kind of classify them. Cancer cells. So cancer is sort of classically non-anchorage dependent. And in fact, that's one of the ways that we actually determine whether they're cancerous or not. We stick them in an in vitro environment where they cannot adhere, and we ask whether or not they die or not. If they don't die and they start proliferating in the absence of these physical cues, then we term them, they are cancerous, determined to be cancerous. Okay, don't worry too much about table one. It's just sort of selected examples of extracellular matrix knockout phenotypes in mice. And I just want to point out a couple of big things, which is that a lot of these proteins, like laminins, fibronectins, collagens, you see the phenotype is lethal. Yeah?
You talk about like putting the cancer cells in a different environment, seeing if they die. Like, isn't that dependent on the immune system actually doing that?
Yeah. So when you think about cancer biology, there are a lot of opposing factors, right? Immune cells being able to recognize them is a whole different topic. And what I'm just trying to mention is that in the case of tumor cells, cancer cells, they do not require physical attachment to an extracellular matrix in order to resist apoptosis and to proliferate. So we put them in something, we do like agarose gels, right? So we'll stick them on agarose that they're unable to attach to. And if they start forming colonies, what we call colony forming units, then they're determined to be cancerous, right? So interestingly, laminin is expressed at the 2-cell stage. When you were 2 cells old, you had laminin. It's the very first extracellular matrix protein to be produced. Do you know when you die? At the 4-cell stage. So you can undergo 2 divisions without laminin before that embryo dies. So kind of trying to demonstrate the importance of the extracellular matrix. Okay. So we're going to talk about the two main biomolecules in the extracellular matrix. The first is that of fibrous proteins. So these are proteins that are polymer forming. They are self-assembling. Yes?
Sorry, what does differentiation mean for cell?
Differentiation is along a specification pathway. Okay, so you can think about it. If I want to make a lymphocyte, right, a white blood cell, those start as stem cells, hematopoietic stem cells, and they undergo a differentiation pathway that generates a mature, what we would call fully differentiated, terminally differentiated, meaning theoretically, and we're challenging that today, theoretically it can't become anything else, right? But hematopoietic stem cells give rise to multiple cell types, right, that are all going down their own differentiation pathway.
Differentiation is just the process from one stage to another?
Correct. Yeah, from one cellular phenotype to another. Usually describing a linear path towards terminal differentiation. But again, the stem cell community would challenge that. All right, so these fibrous proteins, they're proteins that are made and secreted. They might be self-assembling. They might require enzymatic activity in certain cases to assemble. But the point is that they are monomers in what will eventually become a polymer network. The other biomolecule that we'll talk about is that of glycosaminoglycans. And glycosaminoglycans are commonly associated with a core protein. And when they are, they're termed proteoglycans. And we'll tunnel into this. Glycosaminoglycans, the easiest way to remember them is they are repeating subunits of disaccharides, two sugars. Two sugars, two sugars, two sugars, two sugars, two sugars. And it just keeps on going and going. These things can be megadaltons, right? So we've talked about how big proteins are. The average protein size is somewhere around 50 to 60 kilodaltons. These things are multiple megadaltons large. They are very large species. Okay, so here you have the protein species, this cartoon here. The protein species are in green. Proteins like laminin, collagen 4 that we'll talk about here in a minute, fibrillar collagens, fibronectin, and then the Gags, the glycosaminoglycans, things like perlecan that have a protein constituent, but then these repeating sugar units that come off of them. Things like hyaluronin that are completely disaccharides, repeating disaccharides. Decarin, aggrecan is a big one. Things like aggrecan, and we'll talk about them a little bit more, are actually loaded in your cartilage, and they're very hygroscopic, so they love water and they absorb water, and so they allow the cushioning whenever you take a step in your cartilage. So those forces are absorbed by these glycosaminoglycans. All right, so there are two types of matrix. Remember, the basement membrane and connective tissue. We're going to take them one at a time, and we're going to start with the basement membrane. All right, so organization of the basement membrane has three primary locations. The first is that it underlies the epithelium. Again, here you can think about this. These are ciliated, so maybe they're in your gut or maybe in your lung, ciliated epithelium. And again, remember that they're helping to establish that polarity. They have a very specific biochemical nature that tells the cell, that engages the cell's polarity so that it understands what the basal surface is and what the apical surface is. And when you get disruptions in that, you get crazy things happening, like tumor metastasis and things like that. The other location is surrounding cells. So in that case, you can think about muscle fibers. In between the muscle fibers themselves is a basement membrane. Again, that's helping to stabilize the phenotype and stabilize the polarity of those cells within muscle. The other is that they can be in between sheets of cells as well, okay? So you can think about them as being a thin barrier around epithelial, in general, epithelial sheets. All right, four main components of basement membrane. I think it's safe to say when I generate a list, those are pretty easy things to focus on, since I haven't had a chance to blue label things. So the major constituents are laminin, collagen 4, nitogen, and perlecan. And we're going to focus mostly on laminin and collagen, but nitogen and perlecan play important roles themselves. As an assembled sheet, so it's a polymer mesh network that's sort of cartoonized here, and we'll talk about it a little bit more. This forms a sheet that's on the order of 40 to about 120 nanometers thick. Anyone remember how large the average cell is? 10? I heard 10. 10 microns, okay? So this is a few orders of magnitude less, smaller, okay? These are very, very thin, but not as thin as actin filaments, right? Okay. All right, so laminin is a very heterogeneous protein family. 15 different isoforms have thus far been identified, and I'm sure that list is going to continue to grow. It's a very large, very complex protein, and that's going to be a recurring theme in the extracellular matrix. They tend to be large, they tend to be pretty complex. This one is a heterotrimer, consists of an alpha chain, a beta chain, and the gamma chain. And there are multiple alpha chains and multiple beta chains and multiple gamma chains that all mix and match together during assembly into a mature protein that give rise to these various laminin isoforms. Laminin 1 is a good one to remember. Laminin 111, meaning the 111 is the alpha, the beta, and the gamma chain. So this would be alpha 1 chain, beta 1 chain, gamma 1 chain of laminin. Okay? So you'll see them kind of described. Their common names will be laminin 1, 2, 3, 4, 5. But in the scientific community, you might also see people designate them by their alpha subtype, their beta subtype, and their gamma subtype. So it can get pretty complicated. All right. Again, it's going to organize the basement membrane and the key structures and interactions. Okay, so this thing, this is a TEM, where they're actually looking at a process known as rotary shadowing. It's a specialized technique of TEM. Rotary shadowing allows you to look at a single molecule, in this case here. And as you can see, it kind of looks like a cross, right? So it was, I think it's even called biology's cross. And it is comprised, again, of this central alpha chain, a beta chain, and a gamma chain. The important parts are these two short arms, are the beta chain and the gamma chain, and that's where they hold on to one another to form a polymer. So you'll start getting polymer formation off of the beta and the gamma chains. The alpha chain, good grief, the alpha chain can, in certain cases, this N-terminal region, can in certain cases participate in polymerization. But more likely, it's engaging cellular integrins on both, where's my mouse? On both the N-terminus and the C-terminus. And this C-terminal region I'll talk a little bit about here shortly. All right, so it's going to form a polymer. And then what it's doing with its other two ends is it's telling the cell, interact with me with different receptor types. So when I say integrin, an integrin is synonymous with a cellular receptor that's specifically binding an extracellular matrix protein, okay? So it's a special type of cellular receptor. Again, the long arms, the N and C termini of this alpha chain are for integrin and cell binding domains. This is where it engages the cell. All right, collagen 4, it's about 50% of all basement membrane protein. The other 50% is approximately laminin. Perlecan and nitogen do not comprise a significant portion of the mass of the basement membrane. This is going to be, we're going to talk about fibrillar forming collagens. What defines a collagen is this right here. GlyXY repeats. It's one of the first things that you find out. GlyXY, 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 GlyXY. What's interesting is the X and Y tend to be things like prolines. So does anyone remember what proline will do? It kinks the chain, right? So these things have really tight kinks in them, and as a consequence, then they form these very tight, what we call triple helical structures. 3 chains of collagen will come together in these GlyXY repeats to form this tight coil. All right? So it forms a triple helical structure with GlyXY repeats, and that defines a collagen. In the case of collagen 4, we'll compare and contrast this a little bit with collagen 1, which is what we would call a fibrillar forming collagen. Collagen 4 is going to have more interruptions in that helical structure. So GlyXY, X and Y can be any amino acids. They tend to be prolines. When they're not prolines, then you can imagine there's a relaxation in that tight structure, right? And that relaxation makes collagen 4s a bit more flexible than things like collagen type 1. Collagen 4's role in the basement membrane is to provide tensile strength, right? So collagens are very strong in tension and extremely weak in compression, right? It literally is like a woven rope. If I pull on the rope, it's strong. If I push on the rope, it curls up, right, and bends up. All right. Nitrogen is a glycoprotein. We'll talk about what that means in a bit. There are 2 different isoforms. I'm not going to harp on these proteins, except to say that they're important in binding both laminin and collagen type 4. So laminin's going to form a network, and then over here you have collagen forming a different network. And in order to make these 2 networks work together mechanically, you have to have some type of way to cross-link those 2, right? To have them physically connected. How that works is through this protein called nitrogen and also perlecan. We'll do the same thing. Okay? All right, so it's going to bridge laminin and collagen 4 within the basement membrane. Perlecan is a heparin sulfate proteoglycan. All right, so we're going to compare and contrast a little bit later glycoprotein versus proteoglycan. There are differences even though they sound almost the same. But it is a proteoglycan, which means it is mostly sugar structure with a little bit of protein. That's the way I think about it. All right, again, it's going to bind nitrogen, collagen 4, integrins, and heparin. So heparin sulfate, what it's doing is it's connecting that basement membrane to another important molecule known as heparin. Okay, again, bridging laminin and collagen 4. It's going to help make those 2 networks work together as one. So it's like a copolymer. That's the way I think about it. You got one polymer, you're copolymerizing another polymer, and they act independently unless you connect the 2. So these 2 proteins connect those 2. All right. So basement membrane assembly. These are the assembly of the glycoproteins in the Golgi apparatus. They're always assembled in a fashion that we would call, you're going to see me use 2 different types of terminology, and I apologize for that. I've talked to you guys before about something called a pro-protein. Does anybody remember that? What did I say a pro-protein implies when I call something a pro-protein? Yeah. So it's synthesized in an inactive form or in a form that requires additional processing into its mature form. Can anyone think of why, when a cell is making something like collagen or laminin that I've just described as being polymer forming, that's important that it be in a pro form? What's a good guess? Yeah. Yeah, right? I mean, polymerization inside the cell sounds like a pretty bad idea, and it is, right? So when we get defects in the production of the proform, if we start trying to produce recombinantly, let's say, a constitutively active collagen, we actually get the cell filling up, basically dying, because we wind up filling all of the ER and the Golgi with a polymer structure. Right, and that halts those processes. So they're going to be expressed as So when I use this term protomer, I'll go through and I'll change this language in the slides. A protomer is a proprotein. They are synonymous. So I've got some old language in here interspersed with some new language, and I apologize for that. All right, so the laminin heterotrimers and collagen type 4 protomers are assembled in the Golgi apparatus. You see in this case, this is collagen, 3 different chains of collagen, and they're actually assembled within the Golgi apparatus. All multi-chain extracellular matrix proteins, they're translated as individual chains and then assembled within the Golgi apparatus. They're not assembled together. Okay? All right. So then they're going to be secreted. The laminin self-assembles into this honeycomb structure through the interaction with those integrins and another molecule on the cell surface called dystrophin. So the cell is going to, here I've got a vesicle, it's going to fuse with the membrane, and when it presents it, it's going to present it in the presence of collagen and the presence of integrins. The integrins will immediately bind and help to hold the laminin within proximity of each other so it can undergo a polymerization reaction. So what you have is laminin being formed right at the cell surface. Okay? And so those cells, those epithelial sheets that are making the basal lamina or the basement membrane are literally assembling it right up under the surface of their basal surface. It's a little bit redundant. Okay. All right. Collagen 4 undergoes a processing step. There are cell surface associated enzymes in the matrix metalloprotease. Don't worry about that term. There are proteases that are tethered to your cells. And again, as collagen is delivered, right, as vesicles are fusing with the membrane and they're delivered, they bind immediately to these surface associated enzymes which clip the N and the C terminus. So the N and the C terminus are 2 caps you can think about on collagen. They're always expressed with those caps, and the caps prevent them from self-assembling. So the cell's going to spit it out, grab it, and then cut those 2 ends off, and now those collagens, the mature collagen, can now form a head-to-tail polymer. Okay? So this is how the cell is going to regulate their assembly. Again, I'm not going to harp on this, but nitogen and perlecan come in and bridge these 2 independent networks. I do want you to remember that the collagen 4 network is not a part of the laminin network. It's a copolymer system, 2 different polymers that have to be connected physically through nitogen and perlecan. All right. So some interesting stuff. I'm not going to lie. I'm a connective tissue guy, so basement membrane, maybe even I'm a little bit glazy of. There are some really cool tricks that biology uses in processing these proteins. One we did a review on. It's a pretty clever system. So when you cut yourself, your epithelial cells, let's say I have a big old hole. I slide into second base and I've got a big strawberry on the back of my leg, and those epithelial cells have to now detach from their cell sheet and begin to migrate in to fill that wound, right? So what they're going to do is they actually start spitting out laminin in a form that looks like this. Again, I'm going into some details, and I will, if I want you to focus on some of these details, I'll tell you. But the C-terminal end of that alpha chain is comprised of these domains we call LG modules, okay? Laminin globule modules, right? So globular versus filamentous, that's how it got its name. 1, 2, 3, 4, and 5. So it spits it out in this form with 5 LG domains. And in the case of laminin 1, which is the predominant laminin under your skin, that binds an integrin known as alpha 3 beta 1. What you need to know about it is that triggers cell migration. That integrin supports all that cytoskeletal polymerization, membrane protrusion, and cell crawling out over this laminin matrix that the cell is literally spitting out and crawling on, spitting out and crawling on. What's interesting is that there's a proteolytic domain. So this is the first concept of how we're going to get to things like matrikines. These are enzymatic degradation events. In this case, it's catalyzed by an enzyme known as plasmin. There are some other enzymes that will catalyze a cleavage of the LG4 and 5 domains off of this region. Well, that's where the alpha 3 beta 1 binding site was. So the integrin that's supporting epithelial migration into the wound is all of a sudden cleaved off. And now what was hidden and cryptic, so when we talk about cryptic domains, we're talking about domains that are hidden but exposed through some type of processing. There's a cryptic domain in laminin that binds a different integrin, alpha 6 beta 4. What alpha 6 beta 4 does is it says, start making cell-cell contacts, right? So stop, don't migrate, establish firm adhesion to the basement membrane and start making contacts. So it's this really clever time-resolved, it's almost like time-resolved drug delivery, right? So I want X factor for a certain amount of time, and then I want to trigger a change in the cell phenotype. And as a consequence, I'm going to undergo a proteolytic modification of the protein, and now instead of getting the cue to walk, I get the cue to stop and start grabbing a hold of my nearest neighbors. That's why when you cut yourself, you actually don't keep getting your cells migrating over one another, why you don't get large hopefully epithelial tumors, right, growing, right, is that the reason that wounds stop healing when they should is because of these types of time-resolved modifications to the matrix. So you can imagine for tissue engineers, we love this kind of system because now biology's already engineered this time resolution or this sensitivity to certain factors that allow me to engineer now, what if I engineer this site instead of being plasmin sensitive to being sensitive to something that I want to give it. I want to give to a wound. I want to trigger this change at a certain point in time. I can do that. So we tend to like these systems. All right. I'll go way more into a different one with connective tissue that I think is cooler, but that's because I participated in it. So, all right. So connective tissue ECM. We have the basement membrane. Basement membrane's trying to stabilize epithelial sheets, right? Provide support to those sheets, allow them to establish polarity. Matrix is the basal side, the other side is the apical side. In the case of connective tissue, these are always going to be found beneath the basement membrane and or surrounding the basement membrane in things like muscle, right, that are buried. The primary components are these protein fibers, and they're embedded in an amorphous mixture of very large proteoglycans. I kind of did a hit and run on proteoglycans, these very water-soluble molecules, right? So they're water-loving, they're charged, and they're going to hold on to water for all they can, with all their might, right? So if you have a polymer meshwork with water-holding elements inside, then you get a physically robust material. And we'll talk about that. All right, so it's far more plentiful than the surrounding cells. So whereas every epithelial cell might have a basement membrane that it's coming in contact with, there's far more matrix, right? Well, I should have put that the other way. While as the basement membrane will always have sort of a cell associated with it, the connective tissue, you can have large swaths of extracellular matrix where there are no cells anywhere close. Okay? So far more matrix than cells. The connective tissue is what's going to define the tissue properties. So when I pull on my skin, yes, the epithelial cells are giving some resistance. Yes, the basement membrane on those epithelial cells is making sure I don't just pull them off, right? But the actual physical properties of my skin are determined by what we call the dermis, or the connective tissue that's underneath them, right? So the fact that I can pull on my skin only that far is because the collagens are being extended to their extreme lengths, right? As well as elastins that we'll talk about here in a minute. And that's as far as they can go. And if I start pulling any harder, I have to tear all of those that matrix, that material, in order to pull it any further. The reason that when I let go, it goes back is because we have proteins that want to be folded in on themselves, that want to be curled up. Those proteoglycans are making sure that I have good tone to that tissue, that it retakes its original shape. All right, so I've talked a little bit about fibroblasts in the past. These are the predominant cell type in connective tissues. Fibroblasts, we call them the arbiters of tissue remodeling. The reason we do is that they are the main cell that's going to both secrete and assemble the extracellular matrix. They are the bane of our existence in things like fibrosis, where they get a little bit out of control and forget to turn off and keep making matrix and making matrix. You look at any of your scars that you have, and that's a fibroblast that made that matrix and didn't quite know when to stop, right? Things like keloid scarring, all of these types of things are where the fibroblasts have been activated to remodel or to remake tissue that was gone at some point in time, right? All right, so they're going to secrete abundant amounts of extracellular matrix molecules. They are the ones that are going to secrete the bulk of the fibrous proteins. These fibrous proteins will certainly provide strength and organization. And then they will also secrete glycosaminoglycans and these proteoglycans. Again, here we're going to start talking about them. They're highly negatively charged and very hydrophilic, so they love water. These are going to contribute to the hydrated gel-like properties of most of your tissues. And these are also going to resist compressive forces. Right? The easiest example is that of cartilage, right, where you have two impacting rigid bodies like my femur, tibia, right? Okay. All right, so what are Gags? Gags are these disaccharide repeats, and they can be many-fold. The first sugar is always an amino sugar, and the second sugar is a uronic acid. Here's the amino sugar, acetylglucosamine, and glucuronic acid. Repeats of these two disaccharides is what defines things, hyaluronin. Okay, so we define the glycosaminoglycan based on the specific disaccharide repeat. Okay, so that's articulated here. They're distinguished by the type of sugar and the type of linkage between the sugars, right? So this is a different type of linkage than here. Okay, this is like a 1,3 interaction, and this is a 1,4 interaction. Okay. What I want you to know are these four main groups: hyaluronin, chondroitin sulfate, heparin sulfate, and keratin sulfate. Anyone know where keratin sulfate is, or keratin? Yeah. Hair and nails, right? Anyone want to fathom a guess where chondroitin sulfate might be? Yeah. Some in the skin. A lot in your cartilage, surprisingly, a lot in your brain. So when you form scar after you injure your brain, you don't form a scar like in the rest of your body that's comprised of the fibrillar proteins. You form chondroitin sulfate scars, which is kind of crazy. In the skin, yeah. Okay. All right, so the conformation of these proteins. They're very large, extended conformations. They love water. They want to occupy water along their entire surface, and so they will occupy the largest space you give them. So what's going to happen here is that they want to be large, they're highly charged, and so they kind of repel themselves and they want to be linear. So you can imagine if I want to be as linear as I can possibly be, but I'm embedded within a fibrillar matrix, then I'm pushing against all of those fibers. The fibers are strong in tension, and this molecule wants to pull them out. And so what we wind up with is what we call turgor, right? Good tension within the tissue. And so all of your tissues are what we would call pre-stressed. They're pre-stressed because the fibers within your skin within your lungs, within all of your tissues, all of your fibers are always under a small amount of tension because the proteoglycans are confined and want to be spread out. Okay? All right, so they have a very high volume to mass ratio. This is what allows us to form basically a porous hydrated gel, which is what our extracellular matrix is. Functional contributions, the negative charge attracts cations like sodium. They have very strong osmotic pressure. Again, they generate this swelling pressure or turgor. And they withstand extremely high compressive forces due to that swelling. Okay. All right. Chondroitin sulfate, again, just some factoids if you're at trivia night. Chondroitin sulfate's the most prevalent gag. We like heparin sulfate and heparin. Heparin's the highest negative charge density of any known biological molecule. Heparin's really cool because it is in the extracellular matrix and it actually acts as a growth factor storage depot. So a lot of growth factors are charged and a lot of them, most of them are positively charged. Things like fibroblast growth factor. And so what your matrix is doing is as your cells are secreting growth factors, the matrix sucks them up and holds them there, right, so that they don't just diffuse away. And so you have tremendous amounts of local stores of growth factors. And heparin is a big part of that. Hyaluronin is non-sulfated. Again, we're not going to get into the sulfation and the impacts of that, but the sulfation changes the charge. We've talked about how changes in charge change their affinity for one another, change their affinity for water, in this case, change their behavior. All right. Okay, so I only want to get to a couple more slides and then we can hit pause. And then I think hopefully on Tuesday we'll talk about some kind of cool newer science. So I want to address this concept of the difference between a proteoglycan and a glycoprotein. So a proteoglycan is going to contain about 95% carbs, that's the sugar, per weight. So proteoglycan, even though it starts with proteo, which you would think is protein, it's predominantly sugar, predominantly this glycan. Glycoproteins, conversely, are only about 1 to 60% sugar. So it's predominantly a protein, maybe with 1 or 2 sugar moieties hanging off of it. The proteins that comprise the fibrillar network of your matrix are glycoproteins. The different sugars that are added to the predominantly large proteins will actually help also regulate their activity, their affinity for integrin, their ability to bind growth factor, their ability to synergize interactions between multiple receptor systems on your cell. The production of proteoglycans are that there's a core protein that is produced. It's produced in a membrane-bound ribosome, and it moves into the lumen of the ER. Okay, so here is our core protein of any proteoglycan. And then from that, the addition of these glycosaminoglycans occur in the Golgi. Okay, so that's where they're going to be modified. All of that addition occurs in the Golgi, and then they get secreted out. Okay, so what? So you've got a core protein, right? So here's some variations. Aggrecan is a really extreme case of this. Aggrecan aggregates are a core protein of aggrecan with chondroitin sulfate, right? So there's the chondroitin sulfate and keratin sulfate. These are extremely large moieties. Again, megadaltons. And why it's important, I guess I'm going to get to that here in a minute, is that, again, these things want to take on an extremely extended state. And in things like collagen, we're going to restrict their ability to stretch out and occupy their largest space, which is what they want to do with things like, in that case, collagen 3. So we're going to form collagen 3 networks, fibers around these guys, and we're going to restrict them. And they really want to be spread out. So they push extremely hard against that collagen 3. And so when you press on collagen, you're pressing against this tensed aggrecan that wants to be in this conformation and is already being squeezed in. So when you push on it, it'll give a little bit, but it really doesn't want to give a lot. Okay? It's not a rigid body, but it's approaching that. And it's that dampening effect, almost like hydraulics, right? It's using hydraulic pressure almost, in order to dampen the forces between things like your joints. The function, again, it can contribute to ECM assembly and organization, such as decarin. I didn't really talk about that. It can regulate the trafficking of molecules in cells, right? What's interesting about these molecules is that when I'm forming a polymer mesh network, let's say a collagen network, if I have Gags in there, then I can't form my collagen fibers too close to one another either, right? It creates pores. And these molecules can actually be pushed around and moved around by cells. So these structures not only allow tissues to resist compression, they also open up, basically create pore structures that cells can move within. All right, they're going to control, as you can imagine, if they're charged and they're full of water, they're going to control diffusion through the matrix, both through size and charge of that porous network. I mentioned this before, they can bind chemotactic signals during wound healing, right? So they can bind these positively charged growth factors like fibroblast growth factor, which, as the name implies, induces proliferation and growth of your fibroblast population. By binding those signals, they can provide very localized signals during that process. And again, they're going to resist compressive forces, so they're very strong in compression, such as the example of aggrecan and cartilage. Okay, so again, here's a kind of an example of what's happening. These would be a collagen 3 network, collagen 3 fibrils, with aggrecan sort of sandwiched in between. When you press on it, it really does not want to compress any further. When you let go of that compressive force, it's going to re-swell, and that way your tissue, when I push on cartilage, it doesn't stay that way. It goes back to its original configuration. And the reason is that you've got this force that's pushing back out against those compressive forces. All right, we'll talk about the fibrillar proteins on Tuesday, and then, on Thursday. No, on Tuesday, good grief. All right, and we'll talk a little bit more about kind of a cool way that you can use matrix to regulate cell differentiation and morphogenesis of vessels.